Hi, everybody. This is Mrs. Foy, and this is a lecture on Chapter 13 in the Pearson AP Biology book on meiosis. All right, so strap on your seatbelts because this gets really cool, and it's also a little bit confusing because the vocabulary and because there's a lot of similarities with mitosis, and a lot of kids get confused. So we're gonna get through this together, and I think you'll find it very interesting. So when we talk about meiosis, we're just talking about certain cells that go through meiosis. It's not going to be, especially in animals, it's not going to be the majority of cells, just special cells. So I want you to think for just a minute, what would the earth be like if everything that was living today could only go through mitosis and not meiosis, right? So think about that just a minute. Remember, the purpose of mitosis is to form clones. Now, not always what you are forming is a clone, but that's the purpose of it, right? I mean, we do have mutations in the DNA, but the purpose of mitosis is to form carbon copy cells. In meiosis, that is not the purpose. The purpose is to form half the amount of DNA and to have some variation in that DNA. So we'll see it's going to be very different. So genetics, which we're going to be studying next in my class, is the study of heredity and variation. And heredity means the transmission of traits from one generation to the next. And variation just means differences in appearances from parents to siblings. Genes, we know, are units of heredity, and they are made of DNA. And gametes are what we call sex cells. So gametes and sex cells are the same thing, right? These are the reproductive cells. Each gene is found on a specific place on a chromosome called a locus, or loci for plural. And most DNA is packaged into chromosomes, right? So for eukaryotic organisms, it's packaged into rod-shaped chromosomes, but we also have some extra nuclear DNA in eukaryotic cells, and that would be found in mitochondria and also chloroplasts. And the other thing that we know is that in sexually reproducing organisms, we get one set of chromosomes inherited from each parent. And a lot of times on these slides, they'll show those in different colors. All right, so they'll have red chromosomes and blue chromosomes, which are representing the chromosome sets you get from each parent. So here's a great example. Here is one chromosome, right? How do you know that? Well, it's got one centromere here. That's how you count chromosomes. Remember, you count centromeres. So here is a loci where I have a gene, a segment of DNA that happens to code for eye color. And it's a little bit more complicated for eye color. It's not just one gene, but that's okay. And then the trait is our eye color. Here we have another loci for hair color, and then the trait would be what we see. So in asexual reproduction, we have one parent, right? And there's some benefits to that, but one parent is going to give all the genetic information to their offspring. And that is done by a process of mitosis. And usually these offspring of the one parent are clones. Now there can be some differences due to mutations, but basically that is going to be a clone of the parent. In sexual reproduction, we have two parents that only give half of their genetic information to make a unique combination of genes for their offspring. And there's some good and bad things about that as far as evolution is concerned. So let's take a look at some organisms that we know that reproduce asexually. So you might remember when you were a freshman in bio, you might have looked at these little hydra, which are kind of very simple animals that are kind of in the jellyfish category, and they can reproduce asexually by budding. Here are some beautiful redwood trees that we know can reproduce asexually by the roots of the parent tree, which you can see in the center here, kind of sending out, in a way, kind of underground budding to produce these little rings of trees that are called fairy rings. And if you click on that link, you should see this really cool picture looking down on a fairy ring. But all these beautiful, huge redwood trees are genetic clones of the parent. Now we're finding out they're not exactly genetic clones. There is some genetic variability, but that's science, right? We're always finding out cool things. So let's take a look at comparing and contrasting asexual and sexual reproduction. So the pros of asexual reproduction is you don't need to find a mate. You only have one parent, and 100% of the genes, of your genes, if you are an asexually reproducing organism, go straight to your offspring. There's no genetic diversity except that would be found during my mutations. And the con of that is that if you have a harmful gene that's in the parent, it's going to go to the offspring for sure. In sexual reproduction, only 50% of the genes from each parent get passed on to the offspring. But there's genetic diversity because you get a combination of those two. And we're going to see that there are some other ways to introduce variety into the offspring. The cons of that is that you have to go all through that rigmarole of finding a mate and reproducing, and you only get to give half your genetic material. But the fact that your offspring have a more diverse package of genes is very, very beneficial evolutionarily, as we will see. So we need to talk about the difference between somatic cells and sex cells. Somatic cells are just body cells, the regular run-of-the-mill body cells. You know, your skin cells, your hair cells, your heart cells, your kidney cells. They're just body cells. But sex cells are going to be either egg or sperm. And they are very special because they only have half the genetic material. And so what's really important is that you realize that in order to make sex cells, you have to decrease the amount of genetic material by half. That's what meiosis is, all right? And so in humans and in other sexually reproducing animals, we have a sperm that's carries in humans 23 chromosomes, fertilizing an egg cell that carries 23 chromosomes, and they form a zygote. That zygote has 46 chromosomes, and then we go through growth and development through our somatic cells, which go through a whole bunch of mitosis to produce the majority of cells in our body. But in our gametes, in our gonads are ovaries and the testicles produce gametes, which are our sex cells. And that is the only place in a human body where meiosis occurs. So more on that in just a little bit. But let's talk about these other terms, haploid versus diploid. So diploid is what we call the 2N condition. It's the full set of chromosomes. So in a human, that would be 46 chromosomes that we would find in every cell of that baby, with the exception of some sex cells that are under development already in the female, which we'll come to in a minute. So here is our egg cell. The egg cell is going to have the haploid number or N number, and the sperm cells are going to have the haploid number and the N number that are going to come together in fertilization. Now here you can see that these chromosomes were stained pink in the egg and blue in the sperm. How we ever got pink and blue, I don't know, but that's what we do here. And as we're coming together, those are coming together into the first full set of 2N chromosomes. We can see that we have pairs. Okay, so we number our chromosomes in humans 1 through 23. So 1, 2, 3, 4, 5, all the way to 23. The 23rd pair is actually the sex chromosomes, which we'll talk about later. The chromosome number 1 are the largest, and they get smaller and smaller. So actually chromosome 22 is the smallest one, but you have pairs. So here would be, and I don't know if this is this exactly, but let's just pretend. So let's say this is chromosome pair 21, chromosome pair 22, chromosome pair 23, right? These are homologous chromosomes that come in a set in the 2N diploid condition. So somatic cells, we've talked about, are body cells, and a karyotype is what I've just showed you. A karyotype is what happens when a lab technician takes some cells, treats it so that you can see the chromosomes, and basically uses a computer to orderly display them in terms of pairs of chromosomes in a cell. So that's not the way they look, but the technician can do that on the computer so that people can count the chromosomes and make sure that there are a full set. Homologous chromosomes are the 2 chromosomes in the pair, and sometimes we call those homologs. And the chromosomes that are in the homologous pair are the same length, and they carry the same genes that control those inherited characteristics. So here's a technician preparing a karyotype. And so she's looking under a microscope here. She has stained those chromosomes so that you can see them. And then she manipulates the image of those on a computer so that they're lined up by pair. So here's chromosome pair number 1, chromosome pair number 2, chromosome pair number 3, and so on. The 23rd pair is a special pair of chromosomes that may not always look alike. So in the case of a male, we have XY here. If this was a female, we would have 2 X's. So if you look at one pair, and you can blow it up then on this cartoon, you can see we're used to seeing chromosomes as X's. And that's really important that you realize they're not always X's. But here is a chromosome that has been duplicated, has been replicated, and I see 1 centromere here and 1 centromere here. So this is a pair of chromosomes. One of these was from the father, one of these was from the mother, and then I see 2 sister chromatids that are still connected to the centromere. But I'm always counting centromeres, so this is 1 chromosome and that is 2 chromosomes. So when we see a karyotype, we can very clearly see that 22 of the pairs are just regular chromosomes, and we call those autosomes. The 23rd pair is what determines gender, but there are also some other genes on this X chromosome that are necessary for life. So in humans, you can't have a fully functional embryo that has 2 Ys. You have to have at least 1 X because there's other genes on that X chromosome that are necessary for life. So the last pair are called the sex chromosomes, and they can either be XX for a female or XY. So again, just looking at this, here's the British spelling of fertilization. I have an egg, or ovum, plural would be ova, and sperm with 23 chromosomes. They fertilize to form a zygote with 23 pairs. Here's a trick question. I'll ask you again. How many pairs of chromosome are in the egg? 0. Because there's no pairs. There's 1 chromosome 21, 1 chromosome 22, 1 chromosome 23, and so forth. Same thing with the sperm. The only time you get pairs is in a diploid cell. All right. And so, of course, then in the embryo, all we have is just a bunch of mitosis and cell growth. So now let's look at something that we're going to see for the first time now in meiosis. We've already talked about how in mitosis, a chromosome can duplicate during the S phase and make two sister chromatids, okay? And so I would count this as one chromosome and this is one chromosome. But what we're going to see in meiosis is that these chromosomes, these pairs of homologous chromosomes, line up together. They pair up. And so what we're going to see are we're going to see non-sister chromatids, right? So the two red chromatids here are sister chromatids. They should be identical. These blue are two sister chromatids that should be identical. But when we pair up the homologous pairs, we have non-sister chromatids that are going to do something really cool in a minute. And we call this pairing, we actually call it a tetrad. Do you see why? 1, 2, 3, 4. So we have 1, 2, 3, 4 chromatids that are going to be involved in something pretty special in meiosis. So just to keep this straight, in humans and most other animals, the great majority of our cell division is mitosis, right? After fertilization, we're just a bunch of mitosis and growth, right? But the only place where meiosis occurs is either in the male gonad, which is a testes, or the female gonad, which is the ovary. And those produce gametes, which are either sperm or egg. So the only time in most animal development that we ever have meiosis is in special cells in our gonads that are going to produce our sex cells, right? So keep that in mind. But not all living things go through the same, what we call sexual life cycle as humans do. And so I want to start off with animals, which is what we are, because that's familiar to us. But then I'm going to show you some other ways of reproducing sexually that are kind of strange to us because they're not familiar to us. So there are 3 different main types of sexual life cycles. There is one that a lot of protists do. There are ones that most animals do, and there are ones that plants and a lot of algae do. I'm going to start with the animals, then go to the plants, and then talk about the protists and what they do. So in humans, let's always start at fertilization. That's where I think is a good place to start, because we all know that fertilized zygote is going to be 2N. In order to get to the adult forms, all that's going to happen is mitosis, all right? We already have our 2N cell here, and in human form, in the adult form, we just have a whole bunch of 2N cells. But at different places in male and female development, we're going to have meiosis occurring just in our gonads, just in the testes or just in the ovaries. And in meiosis, some special cells that start out as 2N end up being in the N condition when the gamete is fully formed. And so we have haploid gametes, egg and sperm, that are produced. Those then fertilize an egg in sexual reproduction, and we start the cycle over. So let's look right here now, okay? Most of the life cycle of most animals, the great majority is mitosis. Do you see that? And in this diagram here, we see that's in pink, right? So everywhere there is pink, you're going to have mitosis. So I have the great majority of the life cycle, certainly of what we understand in human, is the great majority of our cell division is mitosis, right? The only place we have meiosis is in our gametes for fertilization. That's the only time that you would have an N cell. So again, this should be familiar to you. I always start here because this is what students are more likely to understand. But now we're going to look at what plants and some algae do. And this is a very different way of looking at sexual reproduction, because I can see in comparing this life cycle and this sexual life cycle that half of the cell division is mitosis, but the other half of cell division is meiosis. So that looks really different to us. So we're not going to spend a lot of time. The College Board has cut out a lot, basically everything about plants, which is really unfortunate. But I just wanted to show you that a plant has basically two big life forms. We have something called a sporophyte, which is the adult. So it's the main plant. So you can look out and see a tree or a flower, and the whole plant of the flower, and that would be called a sporophyte. In order to see the actual gametes in flowering plants, you have to look at the flower, which has a female part, which is called the pistil, and the male part, which is called the stamen. And the male produces the male gametophyte, which is a pollen granule. The sperm are in that pollen granule, and it is only, it's the cell that's haploid. The female part of the flower produces the female gametophyte, which is haploid, all right? And we can see that's basically going to be the egg, so to speak. And this is happening in the ovary of the flower, which is in the female part of the pistil. And when fertilization, when this male gametophyte goes down through, it actually grows a pollen tube down to where these ovules are. So they're the in cell here. You have fertilization there, and then you kind of have a pregnancy, so to speak. And that fertilization is going to develop the embryo, which is the seed. And the swelling that's around that is what we call the fruit, all right? And when that seed, which is now 2N, is planted, it goes through a lot of mitosis to form the big multicellular structure that's a tree. So we see in a plant life cycle, where did I tell you to start? Always fertilization. So I'm going to start over here with fertilization. So I have the plant sperm that has the plant sperm is pollen, fertilizing the egg, and I have a 2N zygote right here. And that 2N zygote is going to develop inside the seed, which is inside the fruit. And it's all 2N. So here I have a lot of 2N reproduction that is occurring here. But in the male and female part of the flower, I am producing gametophytes, which are going to be the N cells. So meiosis is occurring here to produce pollen, which is the male gametophyte, and meiosis is occurring here, which is the female egg or ovule. So we call this alternation of generation because about half the life cycle, the organism we call a sporophyte, which is diploid, and the other half is called a gametophyte, which is the part of the plant that produces the gametes. The gametophyte is all haploid, right? So that's producing the egg and the sperm. And so we have this 2N condition, all right? And half the time, the plant is in the 2N condition, and the other half it's in the N condition. So we have these things in plants and fungi and some bacterial called spores, all right? So you might know about fungal spores or fern spores. A spore is actually a N cell that goes through mitosis. All right, and so that's different for us. It's very different, but that's the way that that plants do that. So The sporophyte versus the gametophyte, what's really interesting that we'll talk about when we talk about evolution is that as plants have become more evolved, the gametophyte has gotten smaller and smaller and smaller, and the sporophyte has gotten larger. So like you think about, like a magnolia tree, right? So the sporophyte is the whole tree. The gametophyte is the flower that's on the tree, and it's a lot smaller. The gametophyte's a lot smaller than the sporophyte. When you look at very ancient plants like mosses, we can see that the gametophyte was actually the dominant part of the plant and the sporophyte was smaller. So the sporophyte has gotten larger as plants has evolved, and the gametophyte has been more reduced. And we're going to talk more about this when we talk about evolution, but angiosperms are the most advanced plants. And then the next most advanced are gymnosperms, which are like pine trees. And then, of course, we have ferns, and some of our most primitive land plants are mosses. So again, this is the sexual life cycle of a plant, and I can see the alternation of generation. I see mitosis going on about half the time. And what's different is I see mitosis of cells that are just in. So in humans, we don't do that. But in plants, they have mitosis in cells that become multicellular structures that are just made of N. During fertilization, then, now I get back to 2N, and I can also have mitosis there. So that's different. That's different for plants than for us. So the third sexual lifestyle I want to talk about are the really weird guys. They're not weird, it's just different for us. Most fungi and some protists. And what they do, I can see right away, right? They only have a very small part of their life cycle where they're 2N. The great majority of their life cycle, they're in N, the N condition, and they go through mitosis of their N condition. So let's go to fertilization first, okay? That's how you can get this straight. So in protists are also in fungi. Remember, protists are eukaryotic single-celled organisms. You have these gametes that fuse to form a 2N zygote, but immediately that 2N zygote goes through meiosis to produce more N cells. And those N cells go through mitosis to produce multicellular structures. So that is just another difference in the life cycle of sexual reproducing organisms. So again, these protists, here's some pictures of some of these here. Some of them are multicellular, some of them are single cellular, and a lot of fungi does this as well. So now we're going to talk about mitosis versus meiosis, and we're just going to give a brief overview, and then we're going to focus on the meiosis. So let's look at mitosis first, okay? So again, we have these pink and blue chromosomes, which are significant of chromosomes that came from the mother and chromosomes that came from the father. In mitosis, it doesn't really make any difference because they never pair up. I have duplicated these chromosomes. If they're an X chromosome, guys, they've gone through S. They've gone through DNA replication. So they're replicated, but again, I'm only counting chromosomes. 1, 2, 3, 4, 5, 6. They line up on the metaphase plate, single file, and they are pulled apart by the mitotic spindle, and I end up getting 1, 2, 3, 4, 5, 6 chromosomes, but they're single chromosomes now, not double, right? And so my parent cell is identical to my offspring cell, with the exception that I don't have double chromosomes anymore, I just have single. So I have duplicate, I have a clone of the parent. In meiosis, it's way different. All right, let's look just cursory first. First of all, I have this pairing. Do you see this over here where I do not have over here? all right? So here's the parent cell, and I can see that my chromosomes have duplicated, and I have pairs of chromosomes, a red one and a blue one, that are lining up together. Think of this as like chromosome 1, chromosome set 2, chromosome set 3, where I have one of each. Chromosome 1, chromosome 1, chromosome 2, chromosome 2, chromosome 3, chromosome 3, in a homologous pair. And look at metaphase 1, right? They're lining up in pairs. That is really different. And I see them touching, which is another different thing. And then I have anaphase and telophase, where I get the haploid condition, right? I have 3, 1, 2, 3, but they're double chromosomes. And then I have a whole nother, I'll move this up here so you guys can see, I have a whole nother stage of meiosis where it's going to be just like mitosis. So they're going to divide again, and this time I'm just going to have single chromosomes. So mitosis, think of, I've got identical from the parent cell to the offspring. In meiosis, I am reducing the amount of genetic material from 2N to 1N. A lot of times meiosis is called a reduction division. A reduction division. So you're producing 2 cells and then another 2 cells. So you're producing 4 cells, but it's half the genetic information. So meiosis is very different, and it has 2 stages, and it's going to be only in the gametes, the production of gametes, not somatic cells. So going from haploid to diploid is the essence of meiosis. So here I have a parent cell that has 1, 2 chromosomes. Do you see that? I'm counting centromeres, 1, 2. By the time I get through with meiosis 1, I have 2 cells that have half the number. But do you see, I still have double the genetic material there because I have a chromosome that's an X instead of a stick, all right? So I have to go through another division so that I get 4 cells, each with half the genetic information, and that is when my meiosis is complete. The other thing that we're going to see happen is that we're going to get some combination and exchange of genetic material. That is a huge part of meiosis. It's called crossing over, and it only occurs during meiosis 1. Meiosis 2, if you looked at meiosis 2, and I'll show you this in just a minute, it looks just like mitosis. If you didn't see any of this going on, meiosis 2 looks just like mitosis. So I'm going to start off with my diploid cell in meiosis. This would be a cell that is going to be destined to be either a sperm or an egg. Where is it going to be found? In the gonad, either in the testes or in the ovaries. There are special cells that are 2N cells that are going to go through meiosis and eventually form sperm, or if this was a female in the ovary, it's a lot more complicated in female, but they would produce eggs. So this process in a male is called spermatogenesis. And I'm just showing you an overview right here. Spermatogenesis occurs in the gonads, in the testes, and a 2N cell called a primary spermatocyte is going to go through meiosis and eventually form spermatozoa, which is a sperm. In females, the process is called oogenesis. It occurs in the ovaries, and it actually starts when that female, before she's ever born, which is so interesting. But you can see that in the meiotic divisions, I don't have equal division of the cytoplasm. And so I end up, instead of getting 4 cells, technically I do have 4 here, look, 1, 2, 3, 4, but 3 of the 4 have no cytoplasm and they die. And the 1 cell that has all the cytoplasm ends up being the egg. We'll talk more about that later. So now we're going to dive deep into meiosis. We're going to look at meiosis 1 versus meiosis 2. And we're going to see some very specific differences that we have. Meiosis 1 is where all the action is. Meiosis 1 is the real reductional division, right? So it's where I'm reducing the amount of genetic material in half. Meiosis 2 is another division, but nothing special is happening in meiosis 2. It's just a division of the X chromosomes into sticks. I don't mean X like the X chromosome, I mean the shape of the chromosome. So in interphase, right, during interphase, would I be able to see these chromosomes? No. Remember, it's chromatin. But just to show you a cartoon of what it would look like, I would have my chromosomes during interphase duplicate during the S phase of interphase, and now I have identical sister chromatids that are held together by a centromere. So how many chromosomes do I have here? 2. How many chromosomes do I have in this cell? 2. But they've been duplicated. And now at the end of interphase, before it goes, right as it's going into meiosis 1, they're going to coil up. And I'm going to see them as chromosomes. In this case, we're keeping it simple. And I'm just seeing that I have one chromosome here. Whoops. Those are going to separate, and I'm going to end up with one chromosome, one single chromosome in each of my cells. So here are 3 events that are unique to meiosis, and they all occur in meiosis 1. Something called synapsis and crossing over. So this is where the homologous pairs line up, and they end up touching, their non-sister chromatids touch with an arm of the chromosome on the pair that came from either the mother or the father, and they physically exchange chunks of DNA. That's called crossing over, and that occurs in prophase 1. In metaphase 1 of meiosis 1, the chromosomes are lining up in pairs, not single file. And then in anaphase 1, the homologous chromosomes separate, not the sister chromatids. So all 3 events that are unique to meiosis all occur in meiosis 1. And as I said, meiosis 2 looks just like mitosis. So here we go, right? So this is showing an overview, and we're going to dive deep here. Here is meiosis 1, prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. All the juicy stuff of meiosis is happening here. And then I'm going to have my 2 daughter cells of my original parent cell back here that's undergoing meiosis go through meiosis 2. So we have prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. And as we'll see, meiosis 2, there's nothing special about it. It looks just like mitosis. All right? So let's take a look at what is happening in meiosis 1. So we have 4 stages: prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. Let's look at what's happening here. In prophase 1, so remember, these chromosomes have already been duplicated and they've already been coiled up, so I can see them, right? The nuclear membrane has diminished. And where is this? This is happening either in the testes or the ovaries. And let's focus first on the male, because it's a lot different in the female. So let's say this is happening in the testes. So what I have here is I have chromosomes that are lined up in pairs. So one chromosome, 1 from the mother, 1 chromosome, 2 from the father, are now lined up in a pair, and they actually are touching. The non-sister chromatids, the sister chromatids are identical, right? Because that's what happened in the S phase. But the non-sister chromatids touch each other and their arms overlap. And that is called a chiasmata, because that means X. And you can see that that looks like an X there, right? So the chiasmata is where they're going to overlap, and the chiasmata is where crossing over occurs. And so chunks of the red chromosome are going to end up on the blue chromosome, and chunks of the blue chromosome are going to end up on the red chromosome. Then of course, I see my spindle fiber forming. In metaphase 1, I have my centromere connected to the kinetochore, and here's something else that's different. My homologous chromosomes separate, but the sister chromatids stay together. Okay, so the pairs separate, but the X does not separate into two sticks. That's different. And so let's count chromosomes here. So now I have telophase. I have 1, 2, 3 chromosomes in my daughter cell. How many did I have in the parent cell? 1, 2, 3, 4, 5, 6. Okay, so I've had my first reduction division. So prophase 1 of meiosis 1 is very time consuming. 90% of the time that I have for meiosis happening there. The chromosomes line up, and then we said we have synapsis. So they're going to line up, and then some of the arms may overlap each other. That's called a chiasmata, and they may cross over. Crossing over is when the genetic material in those arms that are touching exchange, and that is happening at the X-shaped region where the arms of homologous pairs, non-sister chromatids, are overlapping, and that's where crossing over occurs. In metaphase 1, the tetrads, right? So these are the tetrad 1, 2, 3, 4. These are the chromatids, 2 from one homolog and 2 from the other, are lined up in pairs. And the spindle fibers, the microtubule spindle fibers, are going to attach to the kinetochore of the chromosomes and pull the pairs apart, not the chromatids. That's important, right? And one of the things that we see here that we're going to talk more about when we have our genetic unit is the way they line up. Okay, so let's look at this just for a second. All right, now again, we've got this cartoon thing going on where blue means chromosomes from the male and red means chromosomes from the mother. So blue means chromosomes that came from the dad, and red means chromosomes that came from the mother. And let's take a look here. I could have these homologous pairs line up this way, and then their destiny, by the way that they line up on the metaphase plate, they could end up, and this is simplified, I could have all the blue chromosomes end up in one cell that would end up making my gametes here. Or, and then of course the other possibilities, I would end up with all red. Or look at the way these line up. Do you see how just by chance I have a blue red and a red blue lining up on the metaphase plate? When these homologous pairs separate, now I have a different combination of genetic information than I would have had over here. And that is called independent assortment. We're going to talk about that later in genetics, but I just wanted to show you that now because this is another source of variety that we get in sexual reproduction. And the whole point of doing sexual reproduction is to get variety. So this is another place where we see that. So here we have prophase 1, here are my chiasmatas. I had crossing over occurring there. My metaphase plate is I've got my pairs of chromosomes lined up, and now they're going to separate in anaphase 1. But the sister chromatids are going to remain attached at the centromere, and they're going to move in a unit to the poles. In telophase, the chromosomes are just being pulled further to the poles by the mitotic spindle, and now I have half the genetic information that was in the parent cell. Cytokinesis follows, and this is where we're going to see a difference, whether this is happening in the testes or the ovary. You're going to have a division of the cytoplasm. In spermatogenesis, to make sperm, the cytoplasm is going to be divided equally, but in making eggs, they're not. All right? So I can see here that I have prophase, this is meiosis 1, right? So I've got prophase 1, metaphase 1, anaphase 1, telophase 1, and now I'm going to start with 2 cells that are now going to be in my meiosis 2. But I can see here in anaphase 1, just want to show you this again. Do you see how that centromere still has 2 chromatids on it, right? So the pairs of chromosomes are separating and they are going to equal poles. So I have reduced the number chromosomes, but I still have double chromosomes because I still have two sister chromatids. What is holding those chromatids together are proteins called cohesins. Cohesins are responsible for cohesion, where the sister chromatids of a single chromosome are staying together in meiosis 1. In meiosis 2, those cohesins are going to disintegrate at the centromere, and you're going to have separation of the sister chromatids, but we'll see that in anaphase 2. So I want to show you this. Here is mitosis. Here is meiosis. All right. So look, in mitosis, these cohesins are going to hold the sister chromatids together until anaphase, and then they disintegrate, and the centromere pulls apart, and now I have 1, 2 chromosomes. Look at what's happening to the cohesins in meiosis. So here is meiosis 1, right? Look, they're holding them together, right? Still holding them together. I can have, there's my chiasmata right there, the X. I have crossing over occurring, but my cohesins are still holding my sister chromatids together in anaphase 1. But in metaphase 1 and metaphase 2 and anaphase 2, those cohesins break down, and the centromere actually now comes apart, and I have 1 chromosome, 2 chromosomes. All right. So in meiosis 2, we can go through this very quickly. I have 4 phases again. There's no interphase in between. No need, right? There's no interphase in between. But in the development of eggs in a female ovary, this meiosis 2 can happen years later. Okay, so that's kind of crazy, but we'll talk about that. So prophase 2, metaphase 2, anaphase, telophase 2, and cytokinesis is happening just like mitosis. So let's look at these. So here I have 3 chromosomes in prophase 1. I'm not going to have any pairing. There's no pairs. The pair's separated, right? They're going to line up single file. The cohesins are going to disintegrate. The centromeres will separate, and now the 2 sister chromatids become their own chromosome in their own right because they have their own centromere. So anaphase, the sister chromatids separate. I have telophase 2 and cytokinesis, and I have 1, 2, 3 chromosomes here, 3 chromosomes here, 3 chromosomes here, and 3 chromosomes here, but they are single chromosomes, not double, and so the amount of genetic material is correct. So in prophase 2, what is happening? The spindle fiber forms, and the chromosomes move toward the metaphase plate. In metaphase 2, they're not lining up in pairs, they're lining up single file just like they did in mitosis, but they're no longer identical. Because of the crossing over that happened in meiosis 1, the sister chromatids are no longer genetically identical. That's the whole point of meiosis, right? Is to make variety. And so now the kinetochores on the sister chromatids that are attached to the spindle fibers, those microtubules are going to shorten. There are non-kinetochore microtubules that lengthen the cell, and they are going to separate just like we see in mitosis. So meiosis, prophase 2 of meiosis 2, metaphase 2 of meiosis 2, lining up single file. Now we've talked about anaphase, sister chromatids separate because the cohesins disintegrate and allow that to happen. Telophase 2, the chromosomes move toward each pole, and the cytokinesis starts to divide the cell. And again, we'll see that if this meiosis is happening in a male, the cytoplasm all divides equally, and each of the 4 cells, which will become sperm, all have the same amount of cytoplasm. But in an egg, they're not. And so we'll talk about that. So what we have now at the end of meiosis 2 is 4 cells, 1, 2, 3, 4, that have the haploid number of chromosomes, but the genetic material is different than it was to begin with because of crossing over and because of independent assortment. So the 3 things that happen in sexual reproduction that allows for variation in the offspring. The three mechanisms are independent assortment, which chromosome ends up on which side of the metaphase plate. When the pairs line up, do they end up like this or this? That leads to the destiny of where they end up in which gamete. Crossing over, where I have hunks of chromosomes on non-sister chromatids from the homologous pairs exchanging genetic material. And then random fertilization. We don't know which, since each egg is unique and each sperm is unique, we don't know which egg is going to fertilize which sperm, and that is another place where we get variation, all right? So independent assortment, we've talked about, happens in meiosis 1, and again, this is just review, and we'll talk about this more later. So when those chromosomes line up, when they pair up, we don't know which side of the metaphase plate they're going to end up in, and so that ends up with different combinations in the gametes, in the daughter cells, just by chance of where they line up. And that can end up with a lot of variability. So the mathematical equation that we use is, we say the different number of combinations when chromosomes independently assort, when they're pairing up in meiosis 1, is 2 to the n, where n is the number of haploid chromosomes. So for humans, our n number is 23, right? Our 2n number is 46. So 2 to the 23 is more than 8 million possible combinations of variety in our gametes, just based on independent assortment alone, not even based on crossing over. Crossing over gives us a lot more variation, because we have pieces of DNA that are exchanged between non-sister chromatids, and that's going to happen during prophase 1 of meiosis 1, and we've seen this before, right? So the non-sister chromatids, the ones that are not identical, the sister chromatids are identical, but when the tetrad comes together, when the homologous pairs form, the arms of the non-sister chromatids can cross over. That forms a chiasmata CDX, and that genetic material can exchange. And so we see that here in this picture. And those chromosomes now are different. That's going to be variety that we didn't have before. And as I go through anaphase 1, the pairs separate. In anaphase 2, the chromatids separate. And I end up with 4 daughter cells. 2 of those are going to be recombinant chromosomes. Those cells are what we call recombinant, because look, one of the 4 is going to have a chromosome that was not involved in crossing over. Here I've got a red chromosome that was not involved in crossing over. But these 2 have, I have seen crossing over. And so that is important, because that is going to be increased variety, those recombinant cells. So here's just a review of synapsis is where they're pairing, chiasmata is where their arms overlap, and crossing over is when the hunks of DNA actually exchange. So recombinant is looking at the chromatids that actually got genetic material from the other homologous chromosome. And what we're going to see from those recombinants is the farther apart they are on the chromosome, the more likely they are to undergo crossing over. So we're going to talk about this more when we talk about genetics, but the closer the genes are, the closer the alleles are on the chromosome, the least likely they are to undergo crossing over. If they are far apart on the chromosome arm, they're more likely to undergo. And so I can see here are some loci of these particular alleles. So I have yellow, white, and miniature. I don't even know what these are for. So maybe it's like, I don't know, some kind of plant cell or something, okay? So I can see that these only have recombination only 1.3% of the time. That means that we can actually produce kind of a rough map because we know that relatively these must be very close together. If the yellow and the miniature the white and the miniature undergo recombination 37% of the time, then we say they are 37 map units apart. So again, we can kind of find out where these guys are. We can kind of make a map of their loci. The yellow and the miniature alleles are the furthest apart. They have recombination 38%, 38.5% of the time, and so they must be further apart. So this is how we can map the loci of where these alleles are on chromosomes, is we can see how frequently they cross over. If they are very close together, they do not cross over very much. If they're on opposite ends of the arms of the chromosomes, then they're more likely to cross over, and that's how we can figure out where these alleles are. So random fertilization, right? So random fertilization is the third way that we get genetic variation, because any sperm can fuse with any egg. So the fusion of two gametes, all right, so remember, each gamete has 8.4 possible chromosome combination from independent assortment. Each of those, when each of those fuse to produce a zygote, we can get any of about 70 trillion diploid combinations. That's not even counting crossing over. So that is the prize of sexual reproduction, is we get a whole lot of variety, and that gives us more ammunition to be able to adapt to our changing environment. That's what gives us the advantage over asexually reproducing organisms. So now I'd like to finish up the lecture on meiosis to talk about specifically spermatogenesis versus oogenesis. So I want you to just look at this first, and you can see, and we're going to start with the male because that's easier, that we have mitosis. We have some cells. These spermatogonium cells are going to be in the testes, and they're going to go through mitosis. So they're just going to make more of them. But these are the cells that are going to be destined to become sperm. And so in the testes, they're going to go through meiosis 1 and form something called a secondary spermatocyte. And then they're going to go through meiosis 2 and form something called a spermatotid. Notice that you have 4 spermatotids, which undergo a little more maturation and form sperm. So they're going to have a head, they're going to have a collar. That collar is where all the mitochondria are that produce the ATP that allows this flagellum to be able to swim toward the egg. In females, we have oogenesis, and we have cells in the ovaries called oogonium cells that are going to go through meiosis 1 and meiosis 2, but these are going to be delayed by many, many years. And they start actually in the very few months of life when that female is still an embryo in her mother. All right, so it's crazy. And we can see that 3 of the cells that are produced at the end of meiosis 1 and meiosis 2 are going to be called polar bodies, and they're going to die. They don't have enough cytoplasm, and that's just a way to get rid of the genetic information and the genetic numbers so we get the correct number. But it's all going to go into this primary oocyte, this oocyte that is going to be the egg. So let's look at the male first, all right? So we have in the testes, we have special tubes that are called seminiferous tubules in the testes. So this is looking at a cross section. This is where meiosis is going to occur, and those reproducing cells are going to become sperm that are going to go into this area right here, which is called the epididymis, and then it is going to go out, and they're going to go out through the pathway that sperm go during ejaculation, and those mature sperm would be ready to fertilize an egg. So I'm going to show you, this is just a couple minutes of a video. It's really, really good. And so I'm going to show you this video and just watch it with you.
In this video lecture, we are going to be studying sperm and their development. Okay, so just starting out with the sperm, and remember that sperm are the male gamete. And also be sure that you understand that sperm are also called spermatozoa. That's the same thing. So if you hear the term spermatozoa, we are referring to sperm. Okay, so let's look at the morphology and what some of the important structures are associated with the sperm. So you have three different regions. There's the head, the midpiece, and the tail. So the head contains a couple of important things, and number one, those are the chromosomes. And so what I'd like to ask you is how many autosomes are there in the head, and how many sex chromosomes? So if you want to pause the video for a moment and think about that, then I'll give you the answer in just a minute. Okay, so remember sperm are haploid, and because they're haploid, that means they're 1N. That means we're going to have 22 autosomes and 1 sex chromosome. Okay, also associated with the head is the area called the acrosome. The acrosome contains the enzymes that are necessary for fertilization, and we're going to come back to the acrosome and how sperm fertilize the egg later. Then there's the midpiece, the next region of the sperm, and the midpiece contains the mitochondria. Why are the mitochondria important for sperm? So if you remember, the mitochondria are the powerhouse for the cell, so that's going to produce ATP. Sperm are motile, and they need the ATP in order to generate that energy to move. The last component of the sperm, or the last segment, is the tail. The tail is called the flagellum. And so this is what the sperm use in order to propel themselves. So when we talk about motility of the sperm, essentially you can think about the flagellum whipping, and its whip-like motion is what propels the sperm or causes the sperm to move. So how are those sperm produced? We're going to look at spermatogenesis. So this is the process of sperm production, how the sperm are actually produced. So keep in mind that sperm production is associated with the Sertoli cells. And remember that the Sertoli cells are associated with the seminiferous tubules. And so where we're going to begin is at the basement membrane, okay? And then you have a Sertoli cell here on the left. You have a Sertoli cell here on the right. And we're going to be talking about spermatogenesis that's occurring between the Sertoli cells. So we're going to start with a germ cell. So the germ cell are the spermatogonia. Germ cells are diploid, 2N. Now, remember that you have two different compartments when it comes to these Sertoli cells. You have what's called the basal compartment. So if you recall, that's from the tight junction toward the basement membrane. And the other compartment is called the luminal compartment. So that extends from the tight junction to the lumen. We're going to start at the basal compartment with the spermatogonia, and as the sperm migrates toward the lumen, they're undergoing development. So beginning with the spermatogonia, males are born with a finite number of spermatogonia. And so the thing here, though, is that these spermatogonia can undergo mitosis. And so they continually, repeatedly divide. They are dividing indefinitely after puberty. In males, they keep producing spermatogonia throughout their life. After they undergo mitosis, then the spermatogonia are going to develop into primary spermatocytes. And they do that by undergoing mitosis. So those primary spermatocytes then, they are also diploid, because whenever a cell undergoes mitosis, the cells that are derived from that are identical. So these have to be diploid cells. Primary spermatocytes then undergo meiosis 1. So when they undergo meiosis 1, they're going to produce secondary spermatocytes. Secondary spermatocytes are haploid, so they are 1N. Secondary spermatocytes then undergo meiosis 2 to become spermatids. And then spermatids undergo a maturation and development stage. So they have to mature and develop to become spermatozoa, aka sperm. So this is your gamete, the male gamete, Okay, this process, spermatogenesis, began with the spermatogonia. Mitosis took place. You produced primary spermatocytes, then meiosis 1 occurred to produce secondary spermatocytes, then meiosis 2 occurred to produce spermatids, then the spermatids matured and developed into gametes, male gametes, spermatozoa. And all of this process was occurring as the cells were migrating from the basal compartment toward the luminal compartment. And once this process is over, the sperm are actually in the lumen of the seminiferous tubule. And I put a little note for here that approximately 50 to 500 million sperm are actually ejaculated, or that's per ejaculation. In this video lecture, we are going to be studying sperm and their development. Okay, so just starting out with the sperm, and remember.
All right, so that was a really great video just to kind of give you the idea of what's happening here in spermatogenesis. So we have a 2N cell in the male testes that is going to undergo mitosis and produce a whole bunch of these. And this starts during puberty of a young boy. And so then those spermatogonium cells go through, become primary spermatocytes, which go through meiosis 1. They are then secondary spermatocytes, which go through meiosis 2. I have these very early spermatids that then mature into sperm. And this is in the lumen of those seminiferous tubules, which are inside the testes. And then during ejaculation, they're going to be released. And this is the functional sperm, right? And so the head of these sperm are going to contain the N number of chromosomes, right? And so a male is going to go through spermatogenesis from puberty until death. In a female, it's very, very different. So the female gonad is the ovary, right? And inside the ovary, we have these little follicles. Follicles are kind of nurturing cells that produce support for the primary ovum. And so females are going to be born with all of these ovum that they will ever have. And they start development during the fetal development. So they are actually, when there is a baby embryo female, the first part of meiosis is actually occurring in her ovaries before she's ever born. And then it stops, it arrests, and then it continues during puberty of a particular follicle that then ruptures during ovulation. So that would happen during puberty. And there is a little more development that goes there, but it is not until the moment of fertilization, if that egg gets fertilized, then it completes meiosis 2. Talk about waiting till the very last minute, right? So I have a real quick little video that's going to show you this.
In this segment of the female reproductive system, we're going to be discussing the ova and its development. What we're going to be discussing when we talk about egg development, we're really talking about oogenesis. So oogenesis is the development or the production of the ova, okay, which is the gamete for females. All right, now oogenesis actually occurs in 3 different phases. There's the fetal phase of oogenesis. There's prior to ovulation, which occurs at puberty. So this is what starts at puberty. And then there's the third phase called after fertilization. So we're going to work our way through each one of these phases. So we're going to start off with the oogonia in fetal development. The oogonia is diploid, it's 2N, and this is considered the germ cell in the female reproductive tract. So the oogonia undergoes mitosis during fetal development. And remember that the oogonia are fixed. The number of those are fixed. And so once the oogonia undergo mitosis, then some of these will actually begin meiosis. So meiosis begins during fetal development. Meiosis may begin, but it's not completed during fetal development. It is only beginning. So during fetal development, the primary oocyte is produced, and that's produced through meiosis. And then that primary oocyte is going to do what's called a mitotic arrest. So the baby has been born, the baby now reaches puberty. At puberty, meiosis 1 is completed. Now let's think about this for a minute. Okay, when I say meiosis 1 is completed, what we're saying is that at puberty, one primary oocyte per month completes meiosis 1. So when meiosis 1 is completed, a secondary oocyte, which if you notice is haploid, and a polar body. Polar bodies disintegrate, so we won't worry about polar bodies. They're not important. The secondary oocyte is what leaves the ovary and enters into the uterine tube. So when the secondary oocyte gets into the uterine tube, one of two things can happen. Fertilization could occur, considering there's sperm in the uterine tube, or fertilization might not happen. If no fertilization occurs, then the secondary oocyte will continue to pass through the rest of the uterine tube, through the uterus, and out the body. If fertilization does take place, now meiosis 2 occurs. And once meiosis 2 occurs, then we're going to produce an ovum. And we get a second polar body as well. Polar bodies, remember, disintegrate, so we won't worry about those. On the oogenesis diagram then, this last path or phase is after fertilization.
I just think that's so amazing, right?
In this segment of the female reproductive system, we're going to be discussing the...
It's just amazing that in the female, remember, a female is born with all the eggs she will ever have. They're actually oogoniums, right? It goes through, it starts in meiosis before while the baby is still in utero. It is going to go through, it arrests until that particular egg is ovulated, and then it's going to go through meiosis, another part of meiosis, right? A secondary oocyte is actually ovulated, and we have our first polar body, which disintegrates. Remember, one gets all the cytoplasm. And then if that egg, that secondary oocyte, is fertilized, then at the last minute, the rest of meiosis is completed, and then we have a fertilized egg with the correct amount of genetic material. Here's my second polar body there that is going to disintegrate. So to end up, natural selection, right? So the process of evolution is resulting in the accumulation of these genetic variations that are favored by the environment, right? So when we sexually reproduce, we have lots of variation, and the variations that are going to benefit us in a certain environment are the ones that are going to get passed on. And sexual reproduction contributes to genetic variation by mutations, but asexual reproduction has that too. But it's accentuated by three things: independent assortment, which happens in meiosis 1, crossing over, which happens in meiosis 1, and random fertilization, which is going to be which egg fertilizes which sperm. So I hope that has been helpful for you guys. I know it's a lot of material, and I'll see you in class.